Thank you, Neil. Thank you for the invitation. I'm going to start with a show of hands. Let's see who has not heard of the term exposome. Who is not familiar? Oh, well, there's only a few actually. So that's more. No, before you saw the title of my talk, raise your hands. Okay, so it sounds like most people have heard the term. I'm going to try and explain to you a little bit about what we mean by the exposome, what are some of its key features, and then especially look at how can we use this word, this concept in our epidemiological studies, and sort of focusing on what type of research questions we can answer with exposomics compared to more traditional epidemiological questions. So starting here, it sounds like most of you are familiar with this, but for those who are not, the exposome was first proposed in 2005 by Chris Wilde, who went on to become the director of IARC, the International Agency for Research on Cancer, and he's a molecular epidemiologist who said, okay, there's been a lot of investment in genetics, in genetic research, and at that time especially amazing tools had been developed to characterize the genome, leading to a huge field of research on genetics. But we hadn't seen the same level of investment or development in the field of environmental exposure. So he said, okay, the genome needs a complement, i.e. the exposome, and that would then represent all environmental exposures, including diet, lifestyle, anything non-genetic, from conception onwards, as a quantity of critical interest to disease etiology. So really, it's a parallel, no? Between we have the genome on the one hand, the exposome on the other hand, and both of these together are important for disease etiology. Now, there's a nice paper that came out last year, really reflecting. This is by Chris Wilde as well, reflecting on the 20 years that have passed since that word was first proposed, and there's been quite a, as you see, quite a rapid increase, especially in the last 10 years, on publications at least mentioning the word exposome. You see the first years were pretty quiet, and then there was more and more uptake. So this is a nice paper for any of you who's interested. It's a reflection on these 20 years. And of course, the first question I think we all ask ourselves is, if you want to measure the totality of environmental exposures, you're never going to be able to do that. And we all recognize that it's not possible. So, and Chris Wilde also stated that in this paper, that although practically unmeasurable in its entirety, there's still a logical case to be made that this entity exists for each individual. And also that even a partial characterization of this exposome would offer major opportunities to understand disease etiology. And also then he went on to say, this is not an argument between sort of what is more important, genes or environment. It's really a way to where we can see both as necessary and complementary. So some characteristics, the main characteristics. If we think of the exposome, the first word I think that characterizes the exposome is comprehensive. So the defining innovative feature of the exposome is its comprehensive nature, encompassing the full breadth of exposures considered over the lifespan. And this is really the characteristic that sets it apart from more traditional epidemiological studies that look more at one exposure, single disease. By nature, the exposome demands more than that. It demands the integration of different types of exposures, including chemical, physical, biological, psychosocial stressors, or at least, some of these different types of exposures, but also their if you can, their interactions, combinations, mixtures. Those are all the type of questions that could be of interest when we look at exposomics. The time dimension is important. Obviously, the exposome changes, is dynamic, as opposed to the genome that doesn't change in a lifetime. So we would ideally also like to capture that time dimension. The same exposure may have different effects at different stages of life. Most environmental exposures have huge temporal variability. And all of these comprehensive features have implications for the measurement of exposure and also for the statistical analysis techniques. We'll come back to some of the statistical techniques in a minute, but I just wanted to say here that just the word exposome has actually given rise to a lot of, it has stimulated a lot of progress in, at least in the field of environmental health, environmental epidemiology. People are working on new technologies to try and capture that comprehensive nature. And these technologies include, for example, and these are just a few examples, the untargeted measurement of environmental chemicals in biological samples, in environmental samples. So this is really a repurposing of the metabolomics methods. These same platforms are now being repurposed to measure environmental molecules as well as the endogenous molecules. So that's what we mean by untargeted high-resolution mass spectrometry, for example. There's plenty of challenges in that, but this is one of the methods that are being developed. Another part is the wearables and personal samplers. So there's developments in trying to develop personal monitoring systems that can really capture multiple exposures at a personal level. And then there's huge developments in the geospatial and remote sensing methods. So this is really building maps that we can then use to assign exposures to location data, so people's residential addresses or work addresses or school addresses. And that is done with satellite data, for example, or land use data and different modeling techniques. A second characteristic is the integration of the biology, information, knowledge about biology, biological responses. Really, if we can integrate exposure, biology, and disease, then that could offer a powerful way to establish better knowledge on biological plausibility and indicate opportunities also to interrupt the disease pathway. So meaning if we can get better mechanistic data, and that might help us to understand better how all these environmental exposures cause their effects. A third characteristic is that the exposome by nature also is discovery-driven. So exposome-wide research really represents a shift from more hypothesis-driven research to discovery-driven research designs. And that's with the idea that it would allow novel and unanticipated associations to emerge by screening in a hypothesis-driven way, in a hypothesis-free way through a large number of different exposures. In that way, it parallels the genome-wide association studies. And that means there's not a strict hypothesis, but that doesn't mean it doesn't have purpose and value. And I'll come back to that in a minute when we go through some of the research questions that we can pose as part of the exposome concept. I think it's also important to see this as complementary. It's not an approach that's going to overtake everything that's done in the very much hypothesis-driven research areas. It's more that this can be a trigger to then perform more hypothesis-driven research. And this could be a reasonable strategy, leading to a comprehensive integration of discovery-driven and hypothesis-driven research designs, which is sort of where we would like to end up. Then those are three main characteristics. The next question then is, what type of research questions can we answer using this as an approach rather than our more traditional approaches? And I want to take you through some of these questions, and I want to illustrate that with an example data set, which is from our own Helix study. That is a study we started way back in 2013, and we built up a fairly comprehensive database. This is based on pregnancy and childhood, so it's an early life exposome study with 3 follow-up points, basically pregnancy, childhood, and now also adolescence. The outcome data is mainly focused on birth outcomes, cardiometabolic health of the children, neurodevelopment, and respiratory health. And in this is not a huge database, as you can see. This is, in total, we have 1,660 mother-child pairs in this database. I think what characterizes it is that we spend a lot of time getting very comprehensive, completely harmonized protocols for the collection of the exposure data and as well as the outcome data. Exposures were measured with different personal monitors and sensors with biomonitoring methods, so measuring concentrations of different chemical pollutants, for example, with traditional questionnaires for variables such as diet and social factors. And then we did a lot of the geospatial modeling to model a lot of the exposures in the urban environment, including air pollution, but also many variables related to the built environment, traffic, noise, etc. And these exposures, we had around 100 different exposures in pregnancy, slightly more in childhood, and these same exposures and the data collection was repeated in adolescence. And then we also added all the different omics layers. Now these are the data layers that we started adding when we started in 2013. Since then we've added some extra omics data also in adolescence using newer untargeted platforms for metabolomics, proteomics, and also the microbiome. So that's the example. And I think here are some of a few quick descriptions of that data. And I think the first characteristic of this type of data set is that the exposures, this whole picture of the exposome, it is highly dimensional. What that means is that there's a lot of interrelationship, a lot of correlation between the exposures. You might have already expected that, but it's really important to really first look at all the correlations and interlinkages between your exposure data before you go on to try and link that to health outcomes. These are the different families of exposures we had, and you can see the closer together these points are, the stronger the correlations. And this is, for example, the whole cluster of urban environment exposures. And you also see that even though most of the highest correlations, the strongest correlations are within families of exposure, there is also a lot of exposures between different families. Now, what that means also, this is one of the first approaches we tried was to try and reduce the number of variables within that whole exposure data set. So we tried to run a principal component analysis, trying to create these latent variables, these principal components, to explain these data sets. But we found that was really difficult. You need a lot of different principal components to explain a good variation in your data set. So that was really quite a challenge. So this basically means it's a complex data set and highly dimensional. A second feature, which is also not surprising is that it's highly variable in time. So the temporal variation of all these different exposures that we measured yeah, there's a lot of variability. We did that giving people sort of daily personal monitors, asking them to collect daily urine samples to really go into trying to quantify the temporal variation. And basically the picture is similar for these urban exposures, for a lot of the chemical exposures, and also for some of our omic signals. Basically they vary a lot from day to day. So that is something to keep in mind when you interpret results from these type of studies. So in terms of research questions, and looking at this highly dimensional picture with lots of correlations, I think one of the questions we can ask is, out of all these exposures that are measured, this is an example data set, there's plenty of other data sets where you might have a lot of different exposures. One question you can ask, which is kind of hypothesis-free, sort of discovery-driven question, is which exposures within that whole mixture of multiple exposures affect the health outcome of interest. And we can do that in a type of analysis that is called the exposure-wide association study. This is in parallel to the GWAS, the genome-wide association study, where you run a single exposure model for each of your exposures. So you have as many regression models as you have exposures with correction for multiple testing. We did some simulation work in the papers quoted here, and we found that actually running these type of XWAS analyses still gives you a very high rate of false positives, even after correction for your multiple testing. And what that means is really when you have highly correlated variables, it's very hard even for this type of exposure-wide association study to pick out the correct, truly associated exposure. So it is recommended to follow these XWAS analyses with the different approaches. And one approach to use here is the variable selection approaches. So these are methods these are algorithms that go through your exposure and pick out the ones that have the strongest, that best predict the outcome using different methods. And these are less subject to these false positives because they take into account the correlation between the exposure variables. Another question we may ask ourselves is, are there specific exposure profiles that affect the outcome? So are there subjects that share similar exposomes, and does that in some way affect health? And this is basically what we do when we do a cluster analysis. We group our study participants according to their exposure profiles. A third question is, are there mixtures or combinations of exposure that affect the outcome? Because if we think of environmental exposures, we're never exposed to just one environmental pollutant or one risk factor. They always come in combinations. And there are several techniques we can use for that. One is the dimension reduction type of techniques, like the principal component analysis that I mentioned before. Another whole set of methods is a method based on mixture modeling, and these are methods that try to create mixture indices related to the outcome, and I'll show you some examples of these. So this is an example of a basic XWAS analysis where each exposure is related to the outcome in single exposure models, and you run that same model for each exposure. And you can visualize that in these type of volcano plots that show the beta estimate and the p-value. So you find your significant findings here, for example. This red line shows the multiple testing corrected p-value. So in this study, there weren't any exposures that were associated with the outcome after correcting for multiple testing, not in the pregnancy, but then when we modeled these same exposures during childhood, we had a whole lot of exposures that were significantly related to the outcome, in this case, the BMI of the child. Some of them in expected directions, some of them in unexpected directions. This is also something you might find when you run these type of models. We're getting a lot of more unexpected directions in our data, which could be, in this case, is probably because of confounding by the adiposity status of the children that interferes with the storage of these lipophilic chemicals. These are chemicals that store in fat tissue. But then we also see some associations, for example, with indoor air pollution and with some other chemicals, with some essential metals, also with smoking of the parents. And in the pregnancy, it's really only maternal smoking. And then in this study, we ran a second step, a variable selection model, an algorithm that gave us these multiple exposure models. So you can see there's a reduced set of variables that are that seem to affect child BMI after correcting for the other exposures. So that's one method. And a different one is the question on the exposure profiles. So here we ask the question of whether exposure profiles, urban exposure profiles, affected blood pressure in children. And this is based on a hierarchical clustering method. The data set here has a larger N because we were able to include more children than in the previous study. And we had here over 100 exposure variables, all related to urban environment exposures. And here in this clustering method, we detected 6 clusters, and the 2 more harmful clusters showed increases, especially in diastolic blood pressure of the children. And these are clusters that were characterized by high exposure to air pollution, to traffic, to noise, and low exposure to green spaces. So these type of methods could be useful to look at similarities in exposure profiles and then linking those to health outcomes. The third example is related to the mixtures. So we can ask the question whether if when we measure a lot of environmental chemicals, for example, we know that people are not exposed to just one. There's a lot of questions around cocktails, exposure to cocktails of chemical pollutants, because they might act on very similar mechanisms. So here the question was, do these mixtures of endocrine disrupting chemicals affect metabolic syndrome risk in children? So here in this study, the mother's blood and urine samples were used to measure a range of chemicals, about 50 different chemical concentrations. And these are chemicals that are related to very much to everyday products that we use, including cosmetics, plastics, food packaging, some pesticides, heavy metals. So we are exposed through many different routes, through diet, etc. And these are of concern in pregnancy. So say we had around 50 exposure variables here, and these were grouped into different families, so based on the structure of the chemicals. And then we ran this Bayesian-weighted quantile sum regression, which is a method that creates, calculates a summary index of a mixture. So it doesn't look at each chemical one by one. And it calculates weights for the relative importance of each chemical in that mixture related to the outcome. And we also ran the single chemical models where we didn't see many associations, but then when we look at the mixtures, we find much more pronounced effects, especially for women exposed to these mixtures of metals during the pregnancy, to these pesticides, and to the two other different groups. You might have heard of the PFAS, the forever chemicals. So these were related to an increased risk of cardiometabolic syndrome in the children. And then there are also several groups that were related to a reduced risk, which is harder to explain. So going through all these exposures systematically for different health outcomes, we start to get a picture of which exposures may be related to some of these different health outcomes. And it's kind of interesting, some of the exposures seem to pop up only for one of our outcomes, whereas others, especially tobacco smoke, this is exposures during pregnancy, seem to be related to several, at least more than one of these outcomes in childhood. So doing this systematically, we start to get a picture of the exposures that might be important for child health. And similarly for these are exposures during childhood. So this is an example of how you can use exposome methods to try and get a more systematic picture and a more comprehensive picture of possible risk factors for these childhood health outcomes. The next question we wanted to ask is, can we actually, apart from just looking at that summary, can we identify statistically exposures that affect multiple outcomes? So this is a logic next question. Instead of doing each study separately in a separate paper, you would try to use all the data we have and try to model the exposome in relation to all the outcomes. Yeah, so that's an extra complication. Again, we started with a simulation study to see what type of methods were suitable for that type of analysis. And the advantage of doing this type of analysis is that you might statistically be able to find the predictors or the risk factors that have an impact across more than one health domain. So those might be the factors that are, that you could prioritize later on when you try to think of prevention or intervention methods. Those are the predictors that would, or the risk factors that would affect more than one health outcome. So this is an example where we ran that analysis with the methods that came out as performing best in our simulation studies, and this is a reduced rank regression. I won't try and explain the statistics, but it is based on jointly modeling these correlated outcomes at different time points, and then looking at the exposure variables in relation to these latent outcome variables. So this takes into account the correlations within the exposures, but also within the outcomes. And you can get some results that describe these sort of master regulators, these exposures that seem to affect more than one health outcome domain. And in this case, we found maternal smoking, as we found in our single outcome studies, indoor air pollution, some endocrine disrupting chemicals, some dietary variables, etc. You can see, for example, here the outer ring represents the cross-domain model, which means the model that included all the different health outcomes. And you see in the pregnancy exposures that active smoking of the mother was one of the main variables that showed an impact across the health domains. Similarly for meat consumption and a few other variables. And this is for the childhood exposures where you see a much more mixed picture, but still some of the same exposures that we saw when we did our single outcome studies also come up as sort of having an impact across health domains. So those are just examples of the type of things you can do when you take these type of exposome approaches, these more comprehensive approaches. as I said at the start, integrating the biological responses is also an important characteristic of exposomics research. And the question we ask here is whether we can identify molecular omics signatures that are associated with environmental exposure. The reason why we would be interested in that is because we would like to better understand the biological mechanisms behind the exposure, disease associations, and also possibly to develop biomarkers of exposure in the past or future disease related to these environmental exposures. So, again, as an example, the same database, and here, this is, again, a discovery, an exposome-wide study linking each of the exposures with each of the omics signatures we had at that time through an exposome omics-wide association study. So here you run models for each exposure with each omics feature. So you run many, many different associations. You then obviously correct for multiple testing. And then you go through different steps to try and interpret the findings. So there's a lot of work on the biological interpretation, testing the robustness, etc. But this is really the aim of this is to screen through all these different associations and build what we call here an atlas of associations, which may then be used in further research to try and replicate that work. So what we did is we built networks of these associations that we found with our exposome omics-wide association. So this is building a network using the betas from the association studies. And this is the pregnancy network, this is the childhood network. And what we found was that when you try to relate the pregnancy exposures with the omics data in childhood, you don't get that many features that seem to be associated with, you don't see that many exposure omics relationships. The other part here is cross-sectional, and what we see there is a highly interrelated network. In pregnancy, again, it's mainly tobacco smoke showing signatures in the epigenetics, in the DNA methylation features, these are quite well described in the literature. And a few other chemical exposures, as well as molybdenum, which is an essential element. So I don't know exactly what that means. And some of the pollutant exposures. And then in the childhood, we see these highly connected networks of different pollutants. Copper, which is also an essential element, pops out. And then several of our chemical pollutants are correlated with metabolites in urine or in serum. So we have quite dense metabolite networks there. And some of the messages, for example, that maternal tobacco smoke shows robust associations and long-lasting effects on the child methylome, both with pregnancy exposure, but also when we look in childhood at parents who smoke and indoor air pollution. We also, as I said, we found very highly connected networks of serum and urinary metabolites linked to these chemical pollutants. And some of that can be explained by dietary intakes, because the same metabolites have been documented as being linked to different dietary sources. For example, fish intake is linked to these metabolites, and some of our pollutants are also, we have intakes of, for example, mercury, arsenic, and PFAS through fish intake. So you see the same metabolites that connect these exposures. So we don't, not all the biological features that we find are necessarily mechanisms. Some of them are more indicators of exposure sources. So this same data can also be used to then look at whether the health outcomes, the exposures and health outcomes are connected through a certain mechanism. In this paper, we looked at the PFAS exposures, and we looked at their association with liver injury measured by liver enzymes in the children. And indeed, the PFAS mixture was quite strongly linked to increases in the liver enzymes that we measured. And we also found several metabolites in the serum metabolome that were, first of all, linked to liver injury markers, but also to the PFAS exposure during pregnancy. So that might indicate some biological pathways. And then a third example is where we use the same omics data layers to try and classify better our phenotypes, in this case, childhood obesity. So this is a way of creating sort of endotypes within a disease outcome, in this case, obesity. So we used sort of multi-omics methods based on machine learning and multi-omics data to define clusters of children defined by these different omics profiles, characterizing sort of more metabolically healthy and unhealthy obesity. And then we looked at the determinants of those different clusters of children, and we found that environmental pollutants like one of the PFAS and mercury exposure identified, they were identified as important determinants of the high-risk cluster of obesity. And then another way to think of the exposome is in the same way that we're building polygenetic risk scores, you might think that in the future we might be able to build exposome risk scores. We're not quite there yet, but we did some first work to use machine learning methods to try and compute these type of prediction scores. So here we're not looking at associations, we're looking at prediction. So we're looking at all the variables together that give the best prediction in the outcome. And we did that for the P factor, which is a combination of behavioral problems, a risk factor, a score for behavioral problems in the children. We had the metabolic risk score and lung function as a marker of lung health, obviously. And what we found is that, we used all the variables available in the database, including omics, all the omics data to try and predict these different outcomes. And we found that doing that, you can predict about 45% of the metabolic risk score, 15% of the behavioral problems risk score, and then only 5% of the lung function. And the feature importance, so the importance of the different exposures was quite different for the different health outcomes. So, for example, for mental health, maternal stress, noise, and lifestyle exposures were important. Whereas for the cardiometabolic health, it was really the protein markers and some features of the metabolome, or quite a few metabolomic features were important for cardiometabolic health. And then a few variables like child BMI and urine metabolites for the respiratory health. So that's another way to think about the exposome and the parallels with the genome is that you can use these type of data to possibly build risk scores. This is only in our own study, so this has not been validated externally. A few take-home messages. The exposome can serve as a framework for more systematic and comprehensive and discovery-focused analyses, as opposed to our more traditional single exposure studies. But even within that, there are many possible research questions, and it's important to formulate those even within these more discovery-driven analyses. Because as I showed you, there's many different ways to tackle these analyses. So you still have a large, a long list of possible analysis methods to choose from. So it's really important, even within that discovery-driven idea, to really formulate those research questions well. The incorporation of the omics data may help to elucidate biological pathways and disease mechanisms, but also you might be able to detect markers of exposure sources. There's plenty of challenges. This is really complex data with a lot of correlations. And these strong correlations between the exposures, what we found in our work also through simulation studies is that it can go both ways. You can get false negatives, which means you don't detect the true associations. Because of those correlations, you might pick the wrong exposure because of the strong correlations. So that you can get false positives. We've seen that. Sample size is important. In all the papers I showed you, I think sample size was a big limitation. As I told you, we have 1,600 children at most in our study, which is not enough when you look at so many different variables. So we really need much larger studies to get to the weaker risk factors. And I think that's also why, maternal smoking keeps popping up, because that's a strong risk factor. And we probably had just about enough sample size to detect that type of increase in risk. But for the weaker environmental risk factors, you really need much larger data sets. And multiple testing correction obviously needs to be done, but then when you're dealing with small sample sizes, you run into problems. We have complex confounding structures, and then all the classical challenges are still important. Missing data, measurement error, but multiplied over many different variables, and all the confounding problems. So I wanted to finish with a little thinking about causality, because even though we're discovery-driven, there's still things I think we can do to get closer to improve causal inference after doing this screening of many variables. And that's mainly through the incorporation of triangulation methods. This is one paper that we published last year where we tried to use some of the triangulation methods to try and get closer to causality in our analysis of mixtures of environmental chemicals in relation to attentional problems in adolescents. Here we measured 30 different chemicals at 2 different time points. We used attentional problems from the child behavioral checklist. And we used single pollutant regression models as a first step. We then modeled mixtures through a parametric G formula. And then we ran instrumental variable regression and Mendelian randomization. First, there's not many SNPs for environmental exposures, as you can imagine. A lot of environmental exposures are not necessarily under genetic control. But we must say that a lot of the biomarkers we measure in blood or urine are still under some form of genetic control, because they are taken up, metabolized, and there's several processes that can be under genetic control. So we could still detect some SNPs for some of these exposures. And then we also tried some negative control methods. And these are the results. To be very short, we found that one of the pesticide metabolites, so indicating exposure to pesticides, was significantly associated with increased attention problems. And this finding was strongly backed by the instrumental variable regression, as you see here. We didn't see much, we didn't find strong genetic evidence in the Mendelian randomization. But the negative control regressions confirmed that we didn't have, residual socioeconomic status confounding was minimal. And another example is this paper from the Alspach group. Also, first starting with an XWAS, and this is using many exposures, hundreds of exposures in the ALSPAC database. So first running the XWAS, all the exposures separately, correcting them for the multiple testing using the Bonferroni correction, adjusting these associations for confounders. So that leaves the study with far less associations. And then trying to replicate that in other cohorts, which is obviously a good step to take for replication. And then also trying to test that in Mendelian randomization studies. And indeed, there were several replicated associations: maternal BMI, smoking again, fat intake, birth weight, previous BMI of the child, but also childhood TV viewing. Some of that was supported by the Mendelian randomization, but not all of these exposures and risk factors, for not all of it was possible to run Mendelian randomization analyses. So there were also some novel associations that could not be replicated or tested in Mendelian randomization. So this is just an example. These are examples of ways in which you can start with an exposure-wide association study and then go through the steps to try and get closer to establishing causality. And in the field of environmental exposures, when we're working with environmental pollutants, environmental contaminants, often that type of triangulation is better done with experimental models. So this is an example from the literature. I was not involved in this work, but here this study starts with a cohort, very similar to the cohort I showed you, where they measured levels of chemicals, endocrine disrupting chemicals, in urine, blood, and they had clinical data, neurodevelopmental data, which was the outcome. They ran a weighted quantile sum regression, very similar to what I showed you before. So they found a mixture of chemicals that was associated with an increase in neurodevelopmental problems, and then they synthesized that mixture of chemicals using these different weights of the quantile sum regression and made this mixture for their experimental work, both in vitro and in vivo, using zebrafish, or I can't remember which models they used. So this is a way to go from the epidemiology to the experimental biology. Starting with sort of exposome pollutant data and then using the mixtures that you get from this modeling in experimental models. That's another form of triangulation. So I want to finish here. I don't know how we're doing for time. Okay. I've gone quite fast, but that's okay. And I thought the main discussion maybe is to see how some of these methods could be used in your fields of epidemiology, because so far it's mainly the exposome has been taken up mainly as an approach in environmental health and environmental epidemiology studies. There's not so much outside the environmental health field. As I said, there's been a lot of development of new methods to measure chemicals, to do geospatial modeling, but all of it is aimed at environmental health. So that's one question. Is this a useful approach in other settings, beyond environmental contaminants? And also, obviously, the your exposome can be very different in different settings. Almost all the work that's been done so far is in higher income settings, in Europe, in North America. So there's a lot of questions also to be asked beyond these high income settings. There's some first examples. There's a nice study from Catherine's team in using data in Mozambique to look at environmental exposures and related to hospital visits. And then there's a big consortium working on social exposome and a lot of different social measures and brain health outcomes and dementia across Latin America. So that's very impressive work. I was hoping to have some discussion on how this type of concepts could be used in your areas of epidemiology as well. I want to thank especially some of my colleagues from ICE Global who have led some of the work I've showed you. And then the whole consortium that worked on these data sets and these papers through the Helix project and more recently through Athlete. And I want to point you towards the International Human Exposome Network, which is a project we're working on right now to build a network internationally to bring people together. And this is also a place where you can find some resources related to exposome research in terms of statistical methods, but also geospatial methods, exposome maps, etc. So I encourage you to look at that website in case you need some further information or further resources. Okay, I will finish there.